Science on Surfaces.
Hello there and welcome to this podcast. In this episode, we will talk about food science. As always, we will start with the basics. So we'll talk about what food science is and when it's useful. And we'll also talk a bit about some recent research in this area. So here with me in this call, I have Dr. Younas Dadmohammadi from the Abbaspourrad lab at Cornell University in the US. And Dr. Dadmohammadi has a PhD in molecular thermodynamics, and he has done a couple of postdoc trainings in energy and food science. His core interest is multi-scale modeling, backboned by extensive characterization measurement, targeting challenges associated with the sustainable food, water, energy, and environment nexus. So welcome, Younas.
Thank you, Malin. Thank you for having me here. So I'm so excited about this podcast.
So great that you could join. I'm really looking forward to this conversation. So you have a lot of experience with food science.
Oh yeah, thank you. I appreciate it.
So could we just start by talking a bit about what food science is? I mean, food, everyone knows what food is, but what is food science? And do we need food science?
That's a great question to start the podcast with. So the food science is, if I want to put as a very simple explanation, food science is a discipline concerned with all technical aspects of food. So beginning with the harvesting or slaughtering and ending to the table, or what we would like to call is farm to fork. And then being more technical about the food science is a kind of it's a cross-disciplinary major which integrates chemistry, biochemistry, engineering, and microbiology and nutrition. The final goal would be better formulation of sustainable process, extendable preservation of the food, and hopefully recyclable packaging of the foods. As a sort of final statement about the food science, so the backbone of the food science lies in an understanding of the chemistry of the food components. So you can talk about the proteins, carbohydrates, fat, and water, and their interactions, and also the reactions that they undergo during the processing, like drying, freezing, pasteurization, canning, and all, of course, during the storage time.
Okay, so how long has this area been around? I mean, how did it start? You know?
So so that's a great question also. So regarding the food sciences is still relatively new and growing disciplines, brought about mainly as a response to the social changes taking place around the world. Food sciences as a kind of knowledge area that not only focuses on our dietary needs, but solves many food-related challenges. It has likely been present in the history of humanity since the beginning.
So it's related to nutrition and storage and like shelf life.
That's correct. Packaging, processing. Yep.
Okay. So like you said, you mentioned it started probably around like the war time,
Yeah.
something like that. Has there been any trends since then, or are we still working with the same challenges, trying to solve similar problems, or are there new areas within food science that have evolved? Or
right. Yeah, it's over the time it's adjusted to the market and the customers' demands. So nowadays we are saying more about the clean label food. So by clean label, I mean the customers can look at the labels of the foods and the market, and they can figure out these ingredients can be found in their kitchen pantry. And then most of these customers, they're looking for the easy to prepare foods, snacks, beverages, and definitely extended shelf life. So these are all poses major scientific and technological challenges to the food stakeholders, not just the customers and the manufacturers in general. And these things can be addressed without looking at the food science as a backbone of this knowledge. And food science understanding, help us to decode the complex biochemistry of the food systems, and knowledge of the methods to process and preserve the foods.
Okay, so you mentioned clean label. What does that mean?
So as I mentioned, so this is a new definition that FDA specifically in US put out there for sort of guidelines. So if you look at the label of the food or the processed food, so you can understand all single ingredients that they used to formulate the food, and then the customer can identify them, or they can have them in their kitchen pantry. So this helps the manufacturer and the new product developer that using some sort of ingredients that they can have a better understanding of the structures, or there is some sources of in a plant and animal basis, more organic types.
Okay, so you're saying that the consumer, they prefer to have more like natural ingredients that they can relate to rather than
Exactly.
some chemical that they don't really know what it is.
That's true. That's exactly true.
Okay, so do you have an example of what could be replaced with a
So
natural ingredient, so to say?
right. So that's a good question, and that's also a huge challenge in the industry facing, because it's the problem. Um, it's not simple as it looks, and so it's lots of challenges you need to consider, considering the pH and the processing, considering the thermal conditions. But let's say the very simple example could be the colorants. So right now, most of the industries, they prefer to use the synthetic color because it has some give some extension to the shelf life. As an example, these synthetic colors are like the red 4D, FDC red 4D, FDC yellow 5, and FDC blue 1. So they can also have the combination of these synthetic colors as a replacement of the natural colorants. But there's a huge demand for them because, as I mentioned before, they have the shelf life stability. But right now, because of these new regulations and like, let's say, customer demands for a clean label and the natural colorant, there's a movement towards the natural colorant. So in our lab, we did lots of a study on a natural colorant. Like one of them is like phycocyanin. The phycocyanin is a brilliantly blue-colored, water-soluble protein. Is a type of proteins, and you can extract it from cyanobacteria. It's such as spirulina, and then. But as I mentioned, this is not a one-to-one replacement. So you need to study and see how you can preserve this natural colorant in a different conditions. For our case, we did a couple of studies on the carbonated beverages, which they have the acidic pH, and we had a challenge to preserve over the shelf life. But we did lots of studies, and now I think we are in a state that we can simply replace the synthetic color with a natural colorant and extend the shelf life.
When you do that replacement, will you get the same blue nuance, so to say, like the same blue color
So
as with
even
the synthetic? Yeah.
right. So the color is pretty much the same. We had the publication that recently published with a collaboration with BioLean and Nanoscience Instrument in the Journal of Food Chemistry. I think we took a lots of good, promising data, I would say. We did a lots of color intensity study, and the color shows the same intensity. And not only that, but over the days of the shelf life, we did some studies over 5 days and 7 days a week under different environmental conditions, that we can preserve this color in a good intensity.
Right. So a consumer wouldn't be able to tell the difference then from a synthetically colored beverage and a natural colored beverage.
That's a goal, even based on the analysis that we did. They prefer a little bit fader color, but a still natural one.
Mhm.
So that's also a positive move towards that directions, if we can totally remove the synthetic colors. Not only that, but replacing the synthetic chemicals with the natural, I would say, molecules. Not only you can replace the synthetic chemicals, but also you eliminate the demand for preservatives. So you don't need the preservative. Most of these natural colorants and the natural flavor, I would say, and natural molecules, they have this capability. They have the antibacterial properties, they have antioxidant properties that they can preserve and eliminate the need for the preservatives.
Okay, wow. So that's two wins basically
Exactly.
with
That's true.
that replacement. Would it be, I mean, probably this is a tricky question, but from a cost perspective, from the manufacturer side, would it be more expensive to use the natural color than the synthetic one?
So at this stage, definitely yes, because right now we are at the infant stage. We're trying to eliminate, so it's not sort of one day problem that you can replace all these synthetic colors. It needs more research and a better understanding of different conditions. But for the case study, specifically for the replacement of the blue color, I think our results shows the promising, and then definitely we need to work on that to bring it to the larger scale of the replacement of the blue color.
So you mentioned there were several different challenges when you are replacing a component, a synthetic component for a natural one. For example, you mentioned the pH.
Right.
So what's the problem with the pH then?
So if you think about the processed beverage or processed food, the manufacturer not only should take care of the preservation of bioactives and the components that they have in the product during the processing. Let's say they washing it with, let's say, very simple steps. They adding a vinegar, they adding different type of acids to adjust the pH, and also that specific components in the food matrix, could be a snack, could be a beverage. They are experiencing different pHs during the process. And that pH could impact the bioavailability and digestibility of that specific components. If we talk about the colorants, that pH could impact the structure of that molecules, and that changes in the structure could either intensify the colorants or faded the color. Not only during the processing, also inside the body, in the digestive tract, in the gut, we are seeing the different range of pH varies from 3 to 7, 7.5, starting from your stomach, which is more acidic conditions, to the intestinal pad. So the component deals with a different type of pHs. So that understanding also helps us to protect that specific chemicals to be survived during different pHs. So pH is a critical factor, directly influencing the, I would say, the preservation and digestibility and bioavailability of these natural components.
Okay, okay. So this is really like it's like engineering work almost.
Exactly, yes. So we need to think about, not only, as I mentioned, during the process, what happened to that specific chemicals, not only in the processing stage, but also during the digestions. When something it's orally administered to your oral cavity, to your mouth, the pH is different. So we need to take care of that. We need to consider to see how that pH could impact the release profile, how the pH could impact the bioavailability of that specific component. So I have a couple of examples that we work on the bioactives specifically. I would be more than happy to discuss about that when I think it's time to discuss it.
Yeah, please.
Yeah, one bioactives that we recently work in collaboration with Bioline and Nanoscience. It's a NAC. NAC, it's a bioactive molecules, which has lots of proven health-related benefits. NAC is the abbreviation of N-acetyl L-cysteine. So it's a bioactive supplement. It's a very powerful antioxidant, and it's used for the detoxification of kidney and liver. But NAC by itself, it has a strong, unpleasant taste. So you cannot just add them to the beverage, or you cannot explicitly administer in oral cavity. So we have a huge problem. The companies, they're trying to have the NAC as a supplements. They have the huge problem with the consumption of just the NAC because of the off taste. So they reached out to us and say if we can use some of the techniques that we are using for masking the off flavor. We propose the encapsulation process that you can think about in a micro scale encapsulation, nano-scale encapsulations. We took that NAC molecules. We're trying to see if we can encapsulate that specific molecules using another molecules. So let's say larger molecules. NAC is a small molecules. We use the protein, specifically in this case, as I mentioned, because we're trying to stick with the clean label standard, we use the egg white protein. So that is also something it's natural, is widely available. And then we're trying to put the NAC inside the pocket of that larger protein. Like, as I said, the ovoalbumin is an egg white protein. So, and then it was the practice, it was successful. So we did lots of sort of characterization tests on that small capsules to see if we can administer the NAC with this in this new micro capsules, and then we have seen that that off flavor it's gone. So you don't feel that much, you don't taste that much that off flavor in that interactions. But the core of that study was how the NAC as a small molecules interacts with the larger molecules like protein, right? So we have lots of characterization techniques in our lab, but one device that we really liked, and then we really take advantage of that device is Qcmd, quartz crystal microballast with dissipations. It's the advantage of that Qcmd that we use is the real-time characterizations. That real-time characterization help us to mimic the oral cavity and mimic the real-time interaction of the ovoalbumin, as I said, is the egg white protein, and the NAC. And that interactions shed light on how we can encapsulate more NAC by using the ovoalbumin.
How do you mimic the oral cavity?
That's a great question. So for the oral cavity, you know, the most of the epithelium inside your oral cavity and inside the body just covered with a very viscous layer of the protein that it's called mucin. So you can use that mucin, coat the surface at the specific pH of oral cavity, which is around 6.8 to 7.2. And then you pass the NAC over, past the encapsulated NAC, past the unencapsulated NAC, over the surface of that coated sensor, I would say, and then see how would be the interactions. Do you see any interactions with the BSM layer of let's say oral cavity? So that's somehow we did this study with other type of small molecules. We did lots of study with the sweeteners, with the sugars, to see how the sugars upon a first sip of drink, how you perceive the sweetness, or how that sweetness linger on your tongue for a longer period of time. by sipping another one, or you're swallowing your saliva again and again, you still see that sweetness. So this is something, this is also a new area for food industry, trying to understand how would be the interactions, how we can better mimic the oral cavity, interacting the mucin and the small molecules.
So basically, you are in this machine. You're coating the sensor with mucin, which is something we have in our mouth,
That's
right?
correct.
So you have a sort of a mouth mimicking surface on the sensor, and then you pass different molecules on top. So you had the NAC and you had the ovalbumin coated NAC and some other molecules, and then you can see how they bind and how long they stay on the surface, right?
That's correct. And then how would be the impact of the pH? Also, this is another impact, yes, and the temperature, because the goal is trying to identify, try to decode the interactions at different pHs, at different, let's say, if you want to use this one as in a hot beverage, what would be the expectation? What would be the release profile? What would be the extent of interaction between these two at different pHs, at different temperatures?
And you want these molecules to interact with the mucin, I guess?
So it depends again. So specifically for NAC, we want to minimize that interactions.
Okay.
Because as soon as interacts, it can trapped inside that layer and then passes through and reaches to the taste receptors, and then the consumer will perceive that off flavor.
Right. So for all the molecules that taste bad, you would like them to not interact with the mucin.
That's correct.
And all molecules and all those that taste good, you want them to interact.
Yes, and then that one is also a tricky part, because if they interact more, and then overall, I would say period of the time, as when you swallow your saliva, you still have that sweetness. This is also, there's a cutting edge there. So most of the manufacturers, they are defining this as a lingering taste. They don't want that much lingering, and that you have too many of these molecules trapped inside the mucin layer, and then it works like a depot. So slowly released to the receptors, and then you can have for a longer time perception of the taste. As I said, it depends to the type of the molecules, but it's a middle line there. Not too much attachments, not very less attachment. It depends to the molecules. Yes, it's interesting.
Yeah, but then I mean, basically, you can then really design the how do you say, like the flavor palette, or like, I mean, you can design how different flavors are not perceived but picked up, I guess.
That's correct. How fast, I would say, how fast you want to something to be perceived, and then what would be or more technical term would be the onset of the perceptions.
Right.
What would be that onset? This is another study that we can do with the Qcmd.
Right, because I think, I mean, just a personal reference, I always think that chocolate flavor disappears too quickly.
Too quickly. Yeah. I've heard about the same complaint about the gums. Yeah, they're saying it's just after 30 minutes of chewing the gum, so you cannot perceive any sweetness. Exactly. But yeah, this is another interesting areas for the food science.
That's a challenge. It's so interesting. So are these the main topics that you're working on, or are you working on something else related to food science at this point?
So our group is pretty much multidisciplinary. We're touching lots of areas, not directly to the food science, also the water treatment. We have lots of projects in the life science, but specifically for the food science, we talk about the bioactives. Another study that we recently submitted to the new journals, it's about the preservation of the vitamin B9, it's folic acid. We know that folic acid is an essential micronutrient with a wide range of the pharmacological applications and lots of good therapeutic benefits such as antioxidants, its anti-cancer, anti-inflammation properties, but it's very susceptible to the environment conditions. For example, if you expose the vitamin B9, folic acid to the oxygen, to the high temperature, acidic or alkaline medium, even light, these environmental conditions impact the stability. And most of the companies, they're trying to compensate for that loss, and then so they enrich the capsules, they enrich the encapsulation, they supplement with more dosage. And we had a couple of good study in that directions to see the impact of the high dose of those supplements for just compensating the degradation during the shelf lives. So we came with the idea that how we can preserve, let's say, folic acid in that process, and then how we can protect the degradation under the light, under the oxygen and high temperature, and as I mentioned, during the range of the pH when you digest it, when you take it orally. We did the same study, and we used another type of protein for encapsulation of the vitamin B9. We used the Qcmd again for the same, for the similar study, try to screen different type of proteins and larger molecules to see how we can put vitamin B9, folic acid, inside the pocket of these larger molecules.
So this would be the same approach as with the NAC and the ovalbumin.
Exactly, but with different challenges, because we have not only the pH and temperature, we have the oxygen, we have the light issues, and also the overall picture would be how we can improve the bioavailability inside the body, and we preserve it over the shelf life. That's the overall picture of these two studies. Yeah.
Wow. Yeah. So how far along are you on that one? Is it still? It sounds very challenging to.
It is. Yes. So we recently, I think, we did with the help of one of our PhD students. She's working directly in this project. She did a great job to get together a very promising preliminary data. We submit this paper recently, and then we are hoping to get published soon, but this is ongoing research in our lab, not only folic acid, but we're touching other I would say health or health supplements, I would say bioactives in that area.
It's super interesting. I was also thinking, by the way, I mean, going back to the previous example that you mentioned where you had the NAC with the ovalbumin, and you're using as a protective material or like a flavor masking molecule, you're using a protein which is good for you, right?
Exactly.
So isn't that's also a benefit?
That's correct. Yes. So we are not only, as I said, so the goal would be how to improve the encapsulation, how improve the shelf life stability, but not using the synthetic chemicals.
Right.
Because you easily can do it. By easily mean it's less challenging compared to the using a natural ingredients. So you can use, you have more options. If you're trying to use the synthetic chemicals for these purposes, you have a shelf full of these chemicals to be used for this purpose, and then the challenges are very less compared to the natural one. But regarding the natural one, the natural ingredients, definitely you need to screen them. You need to understand each molecule separately and the interactions. So that's another area that we are taking advantage of the Qcmd as a sort of the real-time characterizations, trying to screen different candidates for this preservation, for this encapsulation process.
Yeah, it's really fascinating. I mean, so much more to these products that you're consuming than you're aware of.
That's correct. That's
And
because
it's
you're learning.
really engineering work
That's correct.
to design them so that they, behave the way you want to and contain all the nutrients that they need to contain and so on.
That's totally correct. So, and this is not just the food science. We are, as I said, the food science by itself is a sort of cross-disciplinary major. We have in all of these projects, we have a chemist, we have a food scientist, we have a materials engineering team that they are working together to deliver the job to achieve the goal.
Mm. So we've talked about several challenges in food science. What do you think about the future of this area? Sounds like it's here to stay.
Right.
What do you think?
Yeah, so it's a every day it's a new challenge. So we are learning new things. Every single molecules it has its own challenge. So you need to deal with it. But the future, I think, it's going towards, as I said, more clean label food, and then extended shelf life. That's a huge also areas coming. And then by extending the shelf life, you need to figure out to diminish the use of the preservatives, trying to use more natural ones. Create a structures or a platform for these bioactives to be more bioavailable inside the body, have a better digestibility. And also we are part of our lab, they are focusing on lessen the food and agriculture waste. Not only you try to improve the digestion, improve the process, make it more sustainable. And also we have a plan, we have an active project right now to convert the bio-waste, food and agriculture bio-waste to value-added products. We recently kicked off a very interesting sort of research line, try to convert the bio-waste to biodegradable plastics that you can use them for the same in the food science, or somehow we are promoting the bioeconomy in a food and agriculture industry. More specifically, as I mentioned in a previous statements, so we're trying to quantify the in-body and in food matrix interactions between the food ingredients, because you can have small molecules stable at the lab conditions, but in the food matrix, in a snack bar, in the fortified beverage, in the dairy products, you will see a different behavior. So we're trying to understand all these details, not only, as I said, in a food matrix, but also in body interactions. When you digest it, how would be the interaction in oral cavity? How would be the interaction in acidic condition in your gut? How would be in the colon interaction, intestinal tract? How would be that the small molecules interacts and release from? Because you want to encapsulate it, you want to preserve it, but at some point you want that molecules to escape from that cage that you make, right?
Yeah,
You
exactly.
don't want
Yeah,
to preserve it for a longer time. So
yeah.
we had this challenge before for some of the molecules.
Mm.
We did a great job with the encapsulations, but at different pHs we couldn't release that small molecules. So
Mm.
that's the sort of things that we need to think about it, and also improve on based on the specific molecules that we are dealing with. Yes.
Yeah, it's just to me this sounds a bit like nanomedicines almost. Like you're
That's
encapsulating something. I mean, the principle is at least similar,
exactly.
and you want it to be released at the right place in your body.
In your place at the right conditions. That's
In the
correct.
right conditions. Okay, great. So I've learned plenty. Such an interesting area. Super fascinating.
It is. Yes, we're learning every day. So something new. Yeah.
Yeah. So thank you so much for joining us in this conversation, in this podcast. So I think we will end with this for this episode. So thank you for listening to me, Malin Edvardsson, and Dr. Younas Dadmohammadi from the Abbaspourrad lab at Cornell University. And I would also like to take the opportunity to mention to those of you who are listening or watching that if you're interested in surface science and related topics, you should check out our blog, the Surface Science blog. Thank you.
Thanks, Molly.
Thank you.